I want to welcome everyone to the PKD RRC Scientific Roundtable. It's my pleasure to introduce today's speaker, Oleg Palygin. He's a professor of nephrology and the Smart State Endowed Chair of the Renal Disease Biomarkers at the University of South Carolina. I'll just provide a little bit of background of Oleg's scientific career. He got his PhD in biophysics in Kiev, Ukraine, and then came to the United States and did his first postdoc in physiology at the University of Iowa. And then he did a postdoc in neuroscience at the University of Warwick in the United Kingdom. And then he came back to the United States and did a two-year postdoc fellowship in renal physiology at the Medical College of Wisconsin, where he was ultimately promoted to assistant professor. And then in 2021, he was recruited to the Medical University of South Carolina as associate professor and was recently promoted to full professor. During his career, Oleg has developed considerable expertise in a variety of very sophisticated methods for investigating ion transport and signaling mechanisms in the kidneys and in renal channelopathies. Oleg was a recipient of one of our PKD RRC sprint challenges focused on vibrodissection methods, which uses high frequency vibrations to dissociate cells in tissues. So I want to thank Oleg for agreeing to present his work in the RRC, and I look forward to his presentation titled "Leveraging Human and Porcine Tissues to Advance Nephrology Research and Transplant Outcomes." So, thank you, Oleg.
Yeah, I will start sharing my presentation. Just in the beginning, I would like to thank you, the organizers and this group, in particularly Darren and Nicole and Terry, who support our research and help to move our studies forward. And this means a lot for us. And with this, I will start to talk about unique opportunities and unique developed techniques and the methods what we perform in our Kidney Translation Research Center. And if you know, that just quickly in the background, the NIH right now prioritize human research. And we in the kidney area have unique opportunities. We basically not simply can rely on organoids and other stuff. We can produce directly data using human tissue, high quality human tissue. And I will show you right now how many different roads and modalities we can use with human tissue, and how many everything unexplored, and we need more and more research in this area, which is like enormous and have a gigantic opportunities to young researchers and to everybody who want to join the renal field. So I will start with directly with MUC, to just saying why is MUC that in the center and the milestone, particularly for these studies in my career. MUC transplant program, it's really very big in the country. It was number 4 in 2021. Right now, it's even more, and you can see this is around like 410 and donors kidney transplant per year. So this is a really lots of kidney every year. And there is a room not only for the clinical transplantologists, there is a room for research. And I will show you how we elevate these capabilities directly to research and nephrology development. So briefly, organs distributed through the organ procurement partners, which is local for every state. I don't want to stop too much on this one, but we basically have two agreements, contract with, we are sharing Hope South Carolina Organ Procurement Organization with LifeLink Foundation, which is covered 3 states, Georgia, Florida, and South Carolina. So all of these studies approved by We Are Sharing Hope Ethical Committee, and we doing all our research in the direct regulation of Ethical Committee of Organ Procurement Organization, in addition to all IRB and protocols. So just briefly, this center was created by me and Justin, and can be split under three different ways. This is a functional study, tissue biobank, and cell repository. So and I will show you all examples of different these modalities in our setting. So you can look on this website and you can find that information about Kidney Translational Center. And as you can see that the ability of getting transplant organs, it's not associated only with the kidney. We actually can sparkle lots of research in all the area between the kidney and adrenal gland, fat tissue, spleen, vascular studies, and all of these, you can see all of these tissues we collecting, and all of this tissue we put in biobank. We can do the different studies on them, and I will show you some of these examples. So that it's not only about renal research, it's about a connection of renal research to other researcher groups, and really growing in the many directions. So the first question, what is always rising, what the sample distribution and what the patient characteristics we have. You can see here, and I will explain a little bit later why it's particular, these signs indicated here, and why is it particular, these signs represented it more or less. And you can see that around like 78% of our biobank patients are hypertensive, around a little less than 50% diabetes, which is give us very good opportunity to study particularly these two directions at very, very detail. And you see that 30% have no major comorbidities or listed comorbidities from their clinical record. So that's you can see that you can have approximately both ways, diabetes, hypertension, and other diseases if you need to. So you may ask me the major question when you're talking about transplant, why the transplant not used for the people, why it's going to the research. And I will explain you in very brief but detailed how this selection mechanism happens, and you will know in the future how basically the kidney go to transplant or go to research. And the main kidney estimation before the transplant, it's kidney donor profile index. You can go on the website and put your information or anybody information that's just basic, like body weight and history of diseases, age, and serum creatinine, and you can calculate what is a KDPI index. So what is the KDPI index for the kidney if it will be donated? And there is two types of donations what we utilize. First, it's a donation after brain death. This means that there is no warm ischemia on these kidneys, and it's actually represents the best source after life donation. And you can see if it's a patient 33 years old with some of the parameters, no obesity, no history of hypertension, of diabetes, good creatinine. So the KDPI index like 8%. That is just like very, very nice index. And a KDRI index, which just means one, it's a median patient, 63, it means that's basically 37% better than the regular median patient. This kidney have this record. So if you have some comorbidities like hypertension, you can see that's just increased twice your percentage. More important, if you have donation after circulatory death, which expose your kidney to that warm ischemia, probably even a short one. So your index will rapidly change. With the same parameters, you can see that KDPI will be 24%, and with only one comorbidity, the hypertension, is increased to 35. So all of these is just take into account when the kidney will be used for the donor and this is actually reflect how long this graft function of the kidney after transplantation. This is a regular card. What is more common kidney, what we see in the DCD or other people. So you can see there's a 55 years old, no much comorbidities, the KDPI already around 48%. You may ask me what 48% means. So you can see that's very easy chart. You can go on these links. This is for the clinician guides, and it's very simple written. So you can see the KDPI 20%. This means graft function approximately mean time 11 plus years. And when we drop to 86, 100% in KDPI, that is approximately 5.5 years. So it's this really big difference in how how long this organ will work. And sure that there is two type of things what can reject kidney. First, it's just pathology assessment, which is indicate that abnormalities in this kidney using biopsies, or KDPI index, as I explained you KDPI index in the previous slides. So you can see here correlation, and on the bottom you can see kidney donor profile index, and all on the range of from 0 to 85 usually can be used as a transplant. More than 85, this is high risk. And you can see here Rimouzi pathological score assessment, and you can see that the green area, green-yellow area, it's still okay, and the high level is high risk. And you can see that basically even the kidney, we mostly receive the kidney with high KDPI, which is in this range, more than 50. And you can see some of them have very good kidney health, despite the high KDPI, probably because of the age of the patient, or probably that some comorbidities like diabetes, it still could be very good kidney tissue with very healthy healthy status, which will be not use it for transplant and go to our research. We have very pathological kidneys as well. So the both of these assessments give us opportunity to compare the relatively healthy and relatively pathological profiles. So we doing a lot of different pathological scoring using different system and independent pathologists. And you can see here in particularly SETI score, which is shown, and you can see here, this is our typical pathological score. Is basically indicate chronic kidney disease pathologies in the kidney tissue coming to our center. And you can see that approximately 27% of the kidney are relatively healthy, and we have different type of mid changes and moderate chronic changes. So this is represent all the modalities of kidney pathology in our center. So that's we also doing donor kidney assessment score, chronic kidney disease score, and prediction of graft kidney score, NEGFR, for all the patient in our biobank. So this is a typical the card from our biobank, which you can see that how the kidneys biopsy information and patient information looks like. You can see this is a donation after brain death, and with a relatively high KDPI, but still very good scores. It's shown very healthy kidney. And in contrast, you see donation after circulatory death with approximately same high KDPI, but very bad scores, which is shown lots of kidney pathology on the pathology assessment. So it could be both type of the tissue and both type of different patients with known clinical information. So now when I show you brief overview of our kidney center, I will show you how we utilize all of this tissue in three different directions. First, we do human ex vivo in vivo studies. Second is omic analysis, which is one of the most popular direction. And the third one is a clinical interaction and translational research opportunities. And first, I will start with the primary human cell cultures. Primary human cell culture is a very good and very robust tool to study different pathologies, different signaling mechanisms. And we particularly known patient information can produce patient-specific cultures. And here it's just an example of three key investigators, three labs, which is produce different type of culture from the renal tissue. And you can see the Justin van Beskum lab using beads isolation of an endothelial cells culture, which could include a glomerular endothelial cell or renal vascular endothelial cells or aortic renal endothelial cells. And if you in endothelial research, there's all type of modalities of endothelial cells can be, and all the varieties of endothelial cells could be used for your research. And Tommy Noling lab, it's using sieving isolation of the glomeruli to produce mesangial cells culture. And the primary mesangial cell culture from males and females, this is like a key study for the lupus nephritis and diabetes. We all know that mesangial expansion and mesangial matrix damage is related to glomerular disease, but lots of the functional mesangial cells still under study, which is very interesting direction. And my lab is using vibrodissociation technique to isolate bone capsules. And we can grow bone capsule parietal epithelial cells culture, which is rare and a very understudied topic in renal nephrology. So you can see that these methods can produce from the human kidney very unique kind of tissue for any studies. And you can see by sieving, we can produce lots of human glomeruli. You can see how lots of glomeruli from the kidney. This is the human kidney have a lot of tissue. We can produce milliliters of human glomeruli, which is uncomparable with animals like rats or mice. And later, you can produce this into the culture stuff. The vibrodissociation technique that's very nice can be used for isolation of cystic epithelia, but can be used also to isolation nephron segments. And you can see the fine nephron segments isolation when you can separate glomerular, different tubuli, or the bone capsules, like in particularly in the case of our lab, and produce culture parietal epithelial cells. Importantly, we have the ability to isolate different tubules. It could be proximal tubules or cortical collecting duct. You can see here that our publication, which is shown patch clamp electrophysiology in cortical collecting duct. We are able to not only simply record channels and transporter channels response to different stimuli. We can apply different drugs, and this is a test platform for the direct test platform in the human tissue on the different pharmacology. You can see here a basolateral and apical cortical collecting duct examples when we study KIR 4.1, 5.1 basolateral channels and ENaC activity in the presence of different pharmacology, which is published in our paper in the past. Here, it's just very good examples of this vibrodissociation. We can isolate human bone capsule, native, not in a culture, native human bone capsule, load them with different dyes, and we can look different signaling mechanism. And you can see here, we can do this in the rats and in the human. And here, very interesting research, which is shown nitric oxide synthase mechanism regulation in parietal epithelial cells by renin-angiotensin system. There is nothing known about this mechanism, and this is one of the rare evidence how everything this works, and it's really interesting story about parietal epithelial cells physiology. Moreover, we can do electron microscopy, and you can see scanning electron microscopy of the Bowman capsule, which is performed by Daria Latovska in Augusta University. And you can see our Bowman capsules are actually dynamic environment. They're not static sometimes. They very much respond to the disease and inflammation in our body. And you can see this is normal Bowman capsule. You can see these two entrance from afferent arterioles, and you can see this Bowman capsule with entrance of the proximal tubule, and you can see the difference between these two Bowman capsules. They basically show remodeling and pathological remodeling when these cells become from flat normal phenotype to some kind of rocky, and the changing how they look like and how they behave. What it drives this and how this related to the pathology is still unknown. So importantly, we study these mechanisms, and sometimes we see how these cells remodel themselves to something very different. And this is, we always say, is it really the mechanism which we can regenerate human kidney by remodeling parietal epithelial cells to podocytes? This is real million-dollar question, which is still right now exploration of many labs. And you can see these cells, if you stain human kidney from particular this patient, you can see that some of these cells are actually positive to the podocin. So they not only remodel, they develop marker of podocytes, and this is very interesting and fascinating thing. So the more important, we have the tissue, very high quality, and staining in the blocks, which is allowed to do a lot of different things like quantification, immunostaining with all this kidney. Particularly, you can see here our work about ceramide expression in different pathological modalities to see if the ceramides can be, particular long chain ceramides can be used as a biomarker of different pathology. You can see that on this example, very nice staining for podocin, very nice staining from kidney injury molecule and the ceramides and association of these two signalings, so that basically you can use these slides to very high profiles immunostaining and quantification. So the next is omics-driven approach, and you probably know the kidney omics right now. It's an emerged and very popular field of studies, and the human tissue, in particularly different biopsies and so on, usually and most of the time not good enough to perform particular specific metabolomic study, which is required very high profile tissue with very preserved metabolic function, and the best way to isolate from a live tissue and frozen as soon as possible and preserve in the refrigerator for the very short time. And we just writing right now a guideline for metabolomics and kidney function, and we hope this paper will soon be published, and which is actually explain that how you need to choose kidney samples and how for rat and human research, and why metabolomics it's very interesting and different direction type of research which produce a lot of very interesting data. So here I can show you the example of human metabolomics, and you can see this is human renal tissue analysis, which is shown mitochondrial specific metabolome in the human and male and female cortex. And you can appreciate by PCA analysis that human medulla and cortex, in both of this area, males and females are have a distant phenotype, and this phenotype could be attributed to arginine synthesis and purine metabolism. And if you know arginine synthesis, for example, it's a directly nitric oxide production and a lot of mechanism like polyamine and so on. And the purine metabolism just triggering a lot of spark, a lot of different things, including mitochondria function. So all of this is working a little bit different in male and female, and probably all of these are regulated by sex hormones, which we the part of this study. So another study with metabolomics, what I want to show you, it's directly for the polycystic kidney disease profile, which is metabolomic of cystic fluid. In our center, we will be able to collect benign cyst fluid and cyst fluid from the AD-PKD patients. And you can see that profile of this cystic fluid and a difference in this cyst component could actually suggest a lot about the disease progression. They are very different in sense of amino acids and other things. So why that? It's still unknown. And it's very interesting to explore. And Chris Clemens right now pursue this interesting direction in her lab. And last and not least, that we already go into proteomics and use of the tissue. And we usually say just study human hormones just based on the serum hormonal level. But in the tissue, hormonal level could be absolutely different. Plus, in the human exposed to different drugs and so on, human hormone is sometimes not reflect or far away from the hormones what we see in animals. So this is very interesting what we do with Riley Enos from University of South Carolina. This is we build a map of steroid hormones in the different kidney tissues, and you can see how the different distribution between male and female could be in the different tissues. So it's show us a lot of information about hormonal status in particular individual and in particular tissue, and how this reflect disease and phenotype what we see or the predict in our other studies. So and close to the next, I just want to say that since my lab is working in the nephrology division and directly exposed to the clinical problem. We just not staying on the site. We work with a clinician, and this gives us a lot of opportunities to go directly and work directly with not only human tissue, but human plasma patients and so on. Here's a huge amount of different modalities, and I cannot feed all of these in our talk. What we do, I just show a couple of examples which were very interesting in my opinion. So that's this is in particular dialysis, lipophoresis in nephrotic syndrome. It's lots of patients like FSGS patients, sometimes cardiovascular patients just going through this procedure. And you can see that this is standard dialysis procedure, but you have a lipid absorption columns. And you can see we just put the plasma through this cleaning and put it back to the patients. We can collect everything in the bag, everything in the blood, everything going after the filter, and after that analyze. And we can analyze in particular this study related to focal segmental glomerulosclerosis to understand how this procedure actually get a relief for FSGS patient, and how the circulatory component which we can filter in lipid waste can impact the health and progression of FSGS. And you can see this is just a very small example. I'm not going too much. This is just if we wanted to see free fatty acid, we can see we can look on triacylglycerides, we can look on ceramides, but particularly free fatty acid, mostly abundant in the plasma. You can see a lot of them, like palmitic acid and so on, could be elevated in a patient. After lipophoresis, you can see the drastic changes in the profile and rapid decline in most of the fraction of free fatty acids. And we can look not only how they change, we can look how they return back when the patient come into the next appointment. And this is actually can provide us estimation to this patient if this lipophoresis actually help him or not. We can predict this much earlier than the doctor can say, and just stop this procedure after the second time and say this patient have no benefits from this, for example. So this directly implication to the clinical settings of people who exposed to this procedure. One of the main direction of our center is just how I say metabolomics and metabolism. And we know that metabolomics and metabolism of the kidney and different organs implicate in the organ preservation solutions. When we just take an organ from the body and we try to preserve it to donate to another person or transplantation, we put it in different solutions. And one of the most known University of Wisconsin solution to preserve metabolic function. That's what we read in the books. That's what we know from the lots of studies, preliminary studies, and recommendations. But I really check what the changes in metabolism. I really did this study to precisely indicate what kind of metabolites change, how they preserve, how the actual kidney works before and after storage solution. And even that's when we're looking on different solutions, there is 3 different solutions on the market right now. 2 is approved. This is just like histidine tryptophan, which is based on amino acid composition to preserve, mostly using in Europe. In the United States, we use a Wisconsin solution, and there is a new type of the solution which is right now allowed to frozen kidney below the zero and preserve it much longer. This is just a testing solution from external company. We're working with this company and try to explore how the metabolic rates really preserve in the kidneys. So, and here just these studies done with our clinician who just doing transplant studies, and clinical transplant nephrologist and a clinical surgeon who just help us to create all this study and to test how the kidney could be preserved in a different solution and how this impact of metabolic function to develop better preservation, to extend the life of the kidney and delivery of the kidney. And if we go into up to 120 hours, right after that, we have not only local delivery, we can cover worldwide because the kidney can be transferred everywhere on the planet. So, and this is again, this is just when we're using human kidneys, it's very precious tissue, but we can do the same in the pigs. And we particularly work with our DLR and perform some studies in the pigs. So this has allowed us to collect everything what I showed to you, different organs and different tissues in the human. We have this huge collection in the pigs. And why it's important? Because it's just very important for the future xenotransplantation. We should know more about pigs and about their metabolomics, about their kidney function. For example, vasculature function, vasculature in the kidney, in a porcine kidney, is a little bit different from human kidneys. And this is we should take to account when we go to xenotransplantation. We know right now about genes and immune response, but we basically know a little about proteome and nothing about metabolome of porcine kidneys and all about it, how this metabolome and proteome and lipidome develop and function when we put these porcine kidneys in the human body, and how to better preserve porcine kidneys. And this is what we work and in our different experimental settings to try to answer all of these very interesting and highly valuable questions. So overall, I would like to say that our core is just human-centric, and we try to distribute the samples what we collect in, especially for people who want to study and do metabolomics as soon as possible, because how I said, this preservation time really impact metabolome, and especially the specific pathways what people want to look, and the fresh tissue and low storage. It's very important to robust and rigorous research. We serve not only investigators in the South Carolina and Basic Science in VA. We try to collaborate and expand our network And this talk actually kind of like advertisement to join and approach us if you need a human tissue for your research. And as you can see, this could be not only the kidney, and we are happy to help and discuss collaboration. And similar, we're looking on the collaboration on the porcine kidneys and the preservation solution and different analysis. Normotensive renal perfusion right now, very big deal. We developed some stories about this. So all of this, it's required a lot of resources and people, and we cannot do this alone. We need more people collaborate with us to push all of this through. And you can see right now we have, this is not a full list, we have a list of investigators and people who work with our center that cover the different states. And right now we have more and more people join us and taking the tissue for the research. And this program partially supported by American Heart Association and Jennifer Sullivan SFRN project, and we also have our collaborators from SFRN, and particularly Vipul Chitala, who just help us, especially with the right assessment of pathology and navigate us to directly to right conclusion and the treatment of the different kidney diseases. And all of these centers actually help us to pushing this forward. And we have advisory board for sure, which mostly clinicians who just suggest us to, and navigate us in this clinical translational research. We have research project leaders, which is main milestone of our program, which is include Rita, Cannon, and Dasha. And that's the big research. It's just very expensive. That's why we collaborate between two institutions. University of Missouri have one of the best and NIH sponsored PICS facility, and our friend Ade just worked with us on the to develop this external solution preservation project, and as well as our facility and DLR Chris Helke. And this is our team who just working day and night to make this all possible. And if you just talk, which interact with us, and you need any studies in particular, you're probably exposed to all of these guys, which can help you with functional research, culture, or just human tissue sampling. And that's all. That's all my story.
Thank you, Oleg. It was really a nice presentation and a very useful core. Before I get to questions, I wanted to ask a question real quick. As far as the normal human kidneys that you do obtain, do you have a particular cutoff for the KDPI? And do you take into account like warm ischemic time and cold ischemic time? Because I'm sure you're working with a transplant network to obtain these kidneys, right?
Yes, we know all the information about the kidney and their so that's basically we know when the kidney was clipped, the time, and how much time after clipping. We know approximately how long was ischemia, and we know it's just brain or circulation death. And as you can see that organ procurement organization just overview and should take all the best kidneys directly to the patients. So for sure, we're not taking all of these highly valuable kidneys which have low KDPI index. That you see our cutoff approximately 50% of KDPI. And this is, you can say that all our research based on high KDPI kidneys, which as I shown here, it's not always mean that they're pathological. And that's a room for us, for researchers to analyze and work on this tissue and see some pathologies and healthy tissue in particular. So that's yeah, if you ask me, this is a cutoff plus. We just have a limitation. We try to not take in kidney with HIV, hepatitis C, and other very dangerous disease. Try to stay more on the healthy side and so on, which is important due to distribution of this tissue. We try to save not only our staff but our collaborators and just keep them safe. But you always need to treat any kidney tissue like they are very highly dangerous and pathogenic. That's you need to remember, and the lab should be approved. BSL-2 approval should be there to work with a human tissue for sure. Yeah.
Okay. There's a question in the chat. Beautiful amalgam of different parts of the kidney. Are you doing reconstruction of parts for 3D printing type of studies?
No, that's possible to use the kidneys to do the scaffold or to do the just clear imaging, and could be possible to do with a human kidney in our resources. As you can see, we have more than we can chew, and this is a lot of different things. And no, we're not doing particularly this thing. I think this is interesting direction, and if somebody want to collaborate, we're always happy to discuss. Yeah.
So how many specimens do you have in your biobank for normal kidney?
So that limitation right now. This is a funding. We can take more. We have right now we take approximately 3 kidney per month, 4 kidney per month. So that's plus we need the time to characterize and bring all the pathology and clinical information, yeah, which is strictly protected by not only IRB but ethical committee from OPO. So all of these takes a little bit time. Right now we have more than 86 characterized and described kidneys in our biobank, but we have a little bit different banks with the serum and the collaborative effort from Augusta University. If you take only our MUSK-TRC kidneys, which is highly profiled for metabolomics particular study, this is over 86 patients. Which is include complementary tissue of adrenal gland, vasculars, not all of them, but lots of them. Could be include adrenal gland, renal vein, arteria, renal artery, fat deposition, spleen, serum plasma, something else. So that's just not only the kidney tissue, basically.
Does anyone have any questions? Oleg?
This is Madhulika from Kansas University Medical Center. And I was wondering, you have shown the parietal epithelial cells isolation from the glomeruli. Do you also do podocyte isolation, and can they be kept in primary podocytes in freeze in the liquid nitrogen and can be used from there?
So briefly, yes, we do, and we publish this approach together with Sasha Starushenko. We do this in MCW, we do this here. We directly isolate freshly glomeruli, load them with dye, and can image podocytes in their natural settings in the freshly isolated glomeruli, which is, in my opinion, unique and a very useful experiment, much better than any culture and any organoids of anything else. This is just like top level of the podocyte biology and physiology and pharmacology can be studied, in particular these settings. Isolated human glomeruli loaded with diphoridox molecules or for the calcium, and we can look on the podocytes responses on the different drugs. We do particularly that. We just try to do culture things. It's very good, but I'm more like, in my opinion, in particular, freshly isolated tissue. In freshly isolated tissue, like when you take in a tubule, and this is the best what you can have. When you have a human tubule, this is the best approximation to real human physiology. Nothing can be compared. Podocytes as a culture, it's very challenging to because this is cells, it's just challenging to grow. No, and a quick question: no, we not produce podocyte culture. We try to focus on specific things. Unfortunately, podocyte culture not our.
And another question. So what about the vascular smooth muscle cells? I saw that endothelium you can isolate from kidneys. Can also be vascular smooth muscle cells be isolated?
Uh, so that's so that's if we're talking about particularly some kidney cells which have a smooth muscle cells phenotype, my heart is with mesangial cells, because just these cells are actually relative to smooth muscle cells and mediate glomerular hemodynamics, which these cells we grow in and study. As well, we study glomerular hemodynamics, how the glomerulus can contract under different stimulus. No, we not isolate anything else except the cells what I mentioned, because it's I mean, I wish I isolate more different cells, just time and manpower limitation. But we try to build in the specific areas, endothelial cells, mesangial and parietal epithelial cells. Sure, if will be collaborative effort, we would like to explore other things and push more. So like recently, lots of cancer people, I actually need endothelial cells from the lung. So we take in the lung and prepared endothelial primary culture from the lung patients just to help to spark cancer research, for example. That depends on collaboration. I'm always happy to discuss and move something forward and try to.
Thank you.
Thank you.
Awesome. You have a question?
Yeah, quick question. Thanks, Oleg, for the nice presentation. So for the tubules, so you have them frozen, and basically, if somebody want to collaborate with you, we can defreeze them, and then do they grow after that? Or?
No, that's all what I show, that this isolation with vibrodissociation, this is a live tissue. So it could be done, since it's after preservation solution, you need to take the tissue and do a reoxygenation protocol in the piece of the kidney to keep them alive and start moving back their physiological processes on the same level. After that, we isolate defined nephron segments like either glomeruli or specific tubules, and we look on the functional signaling. So this is a live tissue, and it's done the same day when the kidney come to the lab. For sure, we can isolate primary fracture, just primary glomeruli. And with glomeruli, it's very easy. You can see on the screen the glomeruli, due to their physiology, could be isolated pretty precise in a large quantity. When we're talking about specific tubular segment, that's a little bit more challenging, because if you need, for example, cortical collecting duct, or you need proximal tubule, we have these after vibrodissociation setup samples. You need to cut and identify and dissect manually after this isolation. And for the bulk tissue, this could be challenging. You may see that's a lot of manual work. Plus, some tubules could be easy to identify, like cortical collecting duct.
But these you have to do on the same day when you harvest it from the fresh tissue.
Yes.
So you cannot, like, after you isolate the tubules or many of them, like
No,
Cryo-freeze and then recover?
no, they not recover, unfortunately, and the signaling are very. So my lab is study a lot of redox signaling like nitric oxide. That's especially fragile signal, which is tissue lost much quicker than ion channel activity or calcium, and that's required very precise and fast protocols. Yeah, you have short time window, unfortunately. Yeah, you cannot freeze. For the omics approach, yeah, you can freeze and so on. But for functional study, only fresh tissue, yeah.
Okay, thank you.
Thank you.
Any other questions? If not, I'd like to thank Oleg for a wonderful presentation and sharing his work with us.
Thank you very much. Thank you for invitation.